So, welcome to what I understand is the very last of the Greg seminars for this year. We have an excellent seminar today by Haley Carter on the causes and consequences of floral scent diversity in a rare evening primrose. So, Haley did her undergrad at Brown University in biology with a focus on evolutionary ecology, and then moved into a PhD at Northwestern University in Chicago Botanic Garden, advised by Dr. Norm Wickett and Dr. Chris Skogan on floral volatiles and their evolution and genomics in Enithera evening primroses, which is what we're going to hear about today. And round of applause for Haley. Haley defended 3 weeks ago. Excellent. And Haley is now a computational biology specialist at Northwestern, teaching workshops and supporting fellow scientists in plant and biomedical genomics in a role that I think is incredibly valuable in science. So I'm really glad to see Haley continuing in this area. I just want to mention as well that as this is an SSE event, we will be following the SSE Code of Conduct, and a link will be posted in the chat for that if anyone needs to refer to it. So for now, I'm going to turn it over to Haley for what promises to be an awesome and excellent talk. I say this as a fellow floral scent person, but I'm sure it's going to be excellent for everyone. And then we'll have some question and answer at the end. So take it away, Haley. Thanks.
Thanks, Kelsey. So I'm excited to share with you all today what is most of my dissertation research, investigating causes and consequences of floral scent diversity in a rare evening primrose. So just like flowers have a wide and fascinating diversity in color and form, shape, size, they also have really interesting diversity in how they smell. So these images are of three particularly strong or major components of the volatile blend of some characteristically scented flowers. So these volatile organic compounds are the reason that flowers smell so good or so interesting, just depending on what you are experiencing. But floral scent varies not only between, but also within species. I'm going to take one moment to just change something I'm looking at on the Zoom. Okay. So if you took individual lilies, for example, even if most of them produced pretty much the same suite of volatile organic compounds, they might vary in their proportions. Or you might have some individuals in a population that are completely lacking in a particular compound, or others that make compounds that are generally rare across individuals. And when you have this sort of individual variation within a population, we refer to that as a polymorphism. And we might also refer to these different types or morphs of scent profiles as chemotypes. So investigating polymorphisms, how they arise and how they persist in natural populations is a really useful way to dig into mechanisms of evolutionary change. So populations may drift or diverge apart in their traits, scent or otherwise, if they are reproductively isolated from one another, or if they experience different selective pressures. If polymorphic variation already exists in populations, it might persist due to patterns of non-random mating. For example, positive assortative mating, which would be where where individuals of a certain type are more likely to mate with other individuals of the same type instead of those of a different type. This could happen within or between populations and would isolate the morphs reproductively, even if populations are not reproductively isolated from one another. Furthermore, the mode of inheritance of the traits that are variable also changes how we might predict their variation to both arise and persist in natural settings. For example, if you're selecting for a dominant trait, you're selecting for both homozygous dominant individuals and heterozygous dominant individuals because they present as the same phenotype. But keeping heterozygous dominant individuals in the population keeps both the dominant and the potentially non-advantageous allele around. And so you are still going to get variation in your offspring even if you have strong selective pressures for something that's dominant, for example. So I have been investigating all of these potential mechanisms in the plant species Oenothera harringtonii. This is a rare species of evening primrose endemic to Colorado, southeastern Colorado in the USA. It has very strongly scented hawk moth pollinated flowers, and we know that the scent is an important component of pollinator attraction in this system. These hawk moths won't initiate a foraging response or a nectaring response without both olfactory and visual cues. And while Oenothera has a really strong scent across its range, it has really striking geographic variation or a polymorphism for the production of one particular volatile organic compound, and that is linalool. Linalool is a really common floral volatile. You see it in lots of different flowers. If we went back to that image at the beginning of characteristic volatiles in different species, it shows up a couple times. This is a specific enantiomer, so one-handedness version of the compound. And to my nose, it smells like Earl Grey tea or sort of like bergamot, a little bit like the fruitiness of Fruit Loops, if you're familiar with that sort of smell as well. So if you look across the range of Oenothera harringtonii, this map shows on the background elevation. So the redder colors are higher, the greener colors are as you move east into the sort of open prairies of southeastern Colorado. And then the populations are shown in the black dots and then labeled with a color corresponding to the proportion of linalool producing plants in that population. So you can see in the northwest of the species range, linalool is really dominant. It's the dominant chemotype. Almost all individuals produce linalool. They happen to produce a lot of it as well. And then if you're out here in like Comanche National Grasslands or other areas of southeastern Colorado, you don't see almost any linalool production in any of the individuals in these populations. And then sort of along Highway I-25 here, we have this interesting clinal variation zone where it's really variable in any given population how many individuals have that linalool scent or not as part of the blend of their floral fragrance. I might refer to these different chemotypes, linalool producing as Lin plus, non-linalool producing as Lin minus, slightly less of a mouthful. And I'm using the system again to investigate those sort of mechanisms of change that could help explain why or how we got this pattern of variation and if it might persist into the future. So to sort of group those into themes, that would be looking at connectivity of the populations and of the chemotypes, the potential of selection to be driving this variation that we see, and then the inheritance of linalool and a couple of other compounds to see if that can help explain some of these patterns. So for connectivity, the aims here are to quantify long-distance pollen dispersal between populations, to test for differences in rewards between chemotypes that might help explain patterns of assortative mating if they exist in this system, and to test the ability of hawk moths to learn a foraging preference for linalool-producing plants. In the selection section, I'm exploring geographic variation in potential selective pressures, focusing specifically on insect interactions. So interactions with a pollinating insect and a predating insect. I'm testing the impact of linalool on these interactions in the different populations and in these different sort of regional areas. And then calculating selection coefficients for linalool across populations using measures of realized and potential fitness. Finally, I've been measuring the heritability and identifying the mode of inheritance of linalool production and comparing that to other terpene compounds in Oenothera harringtonii. And then I've tested for associations between linalool production and genetic variants using a genome-wide association study for this species. So let's start with connectivity. For this section, I'm focusing on that clinal zone where linalool production is really variable. And I'm including data from 5 populations. With a focal population, RAU or ROUSE in red here. And this population tends to have a 50-50 split of linalool producing and non-linalool producing plants. That is what the B panel here shows. So each individual circle is an individual plant in ROUSE. And the gray is Lin plus, white is Lin minus. And so if you see clusters of gray, you get a little darker. And so you can see this is sort of distributed throughout the population. So what I've done here to look at interpopulation mating and potential for assortative mating is collected leaf tissue, floral scent, and trait data on all reproductive individuals on all 5 of these populations in one spring. This is an annual plant, so we're looking at 1 year of data. I used microsatellites to genotype all of those plants, and then I germinated and genotyped the offspring from the maternal lines in Rouse. And I assigned paternity using those genotypes to those offspring. And then with the trios, I can calculate within and between population mating rates because I have all of the mating pairs. I also was interested in the potential for Hyles lineata is the hawkmoth pollinator in the system, to learn to forage with a preference for linalool. So we know that these moths don't have an innate preference for this chemotype. However, the hypothesis was that if they have only experienced this highly attractive flower in the part of the range where linalool is a really strong part of the sort of signal, they might learn to forage with a preference for that and bring that to other parts of the range. So we allowed moths to forage on linalool augmented plants, have a really rewarding experience with that. And then we flew individuals in a wind tunnel with a choice between a non-linalool producing Oenothera harringtonii individual and a non-linalool producing Oenothera harringtonii individual, to which we added the amount of linalool plants tend to make. All right, so from the paternity assignment portion, I was able to assign paternity to 105 offspring from 75 different mating pairs. 45 of those mating pairs included a pollen donor from outside of that focal population, Rouse. So in this figure, what we have is the pollen donors grouped by their population of origin. And then the maternal lines here, the height of them is just the number of offspring they produced. And then these flows are individual offspring connecting the pollen donor to the maternal plant. Maternal boxes that are white received pollen from both plants within the same population and plants from other populations. Maternal plants that are light gray only received pollen from pollen donors outside of the focal population, and the dark gray only received pollen from pollen donors within the population. So you can see there's a really considerable amount of inflow of pollen from the surrounding populations within Rouse, which shows that pollen is regularly being dispersed the 10 to 32 kilometers apart that these populations are. Great. I also compared floral traits of all of these different parental plants and found that across populations, the traits, including things that would be rewards for the pollinators, so nectar or nectar sucrose, did not vary between the 2 chemotypes. Within Rouse is the B panel. I did find that we saw slightly larger corollas and filaments in Lin plus plants, but there was still no difference in traits that would be rewarding. So nectar and sucrose are not significantly different between the chemotypes. And then working directly with the moths, we saw no significant difference in choice between linalool augmented or non-linalool augmented plants in the wind tunnel. There was also no difference in the time to choose, and it didn't matter if these were male or female moths. So taken all together, this first section shows that this system should be really well connected. We don't have any evidence of isolation that would drive either genetic drift or allow for easy divergence between populations. In fact, almost half of the pollen donors recorded in this study were actually outside of our focal population. And we also have no evidence that hawk moths would learn to forage with a preference for the Lin plus plants, or that the chemotypes are differentially rewarding in a way that would also maybe drive hawk moth preference. I think it'd be interesting potentially to do this with the other populations as well. I have all of the same data for the populations in addition to Rouse. So it might be interesting to look at pollen flow between populations as a way to look at hawk moth movement across the landscape. We don't totally have a great understanding of if they sort of move directionally or if there's a pattern to how they're interacting with these populations. And there's also potential in the system to test for pollen dispersal at even greater distances. 32 kilometers is the previously reported sort of maximum of hawk moth pollination dispersal, but there are obviously more populations here of Oenothera harringtonii, so there's a potential to look for movement beyond 32 kilometers. All right, so looking at selection here, I aimed to explore variation in potential selective pressures, focusing on insect interactions. So looking at very geographic variation of those insect interactions, and then looking at the effect of linalool on interactions in different populations in the sort of different contexts where linalool is rare or common or really variable. And then I calculated selection coefficients for linalool across these same populations. All right, so I've introduced a bit Hyles lineata, which is the primary pollinator of Oenothera harringtonii. However, in the larval stage, it is also a herbivore. These little caterpillars, actually, they can be quite large. Caterpillars will munch on the leaves, the flowers, the flower buds. They can do some damage, but Harringtonii is pretty well chemically defended, actually, against these particular larvae. So a potentially more influential herbivore in the system might be Montha definitella. This is a specialized seed predator. Monthas specialize on Oenotheras particularly. There's sort of a cross-genus relationship going on there. And Montha definitella adults, we believe, visit these open flowers. They crawl down the floral tubes and oviposit so that their larvae develop with the developing fruits, and then they will eat all of the seeds that are developing in the carpel in which they are laid. So they can be kind of a pretty devastating predator as they can reduce your reproductive output by a fourth if you have one, or half if you have two. So it depends on the strength of the infestation there. So across these different populations in the linalool zone, non-linalool zone, and clinal zone, as I'm going to refer to them, we measured pollinator visitation and herbivory, including the number of visits per hour of both hawk moths and bees. Bees are not a very effective pollinator in the system, but they visit, so we scored them as well. We also recorded the proportion of plants with Hyles lineata caterpillar damage in the population, and the number of Montha definitella exit holes per fruit per plant per population as well. To look a little bit more directly at the effect of linalool, we did some experiments where within the non-linalool zone, we added linalool experimentally to a subset of plants. And so then we had a control treatment, a linalool augmentation treatment, and we measured pollination and herbivory on those plants. In the linalool zone, you can't take that away. So instead, what we did was transplant plants from the non-linalool zone up to the linalool zone and set out an experimental array with non-linalool producing and linalool producing plants, where again, we sat and recorded pollinators, and then we, after the fruits were matured, scored Montha definitella infestation as well. Then in the clinal zone, we directly measured differences between Lin plus and Lin minus plants because we chemotyped pretty much all of the individuals in all of those populations over the course of 1 year. And those differences again are in fruit set, flower set, fruit set, and then Montha definitella. So in each of these cases, both where we're adding linalool, where we have an experimental array, and where we're directly counting flowers and fruits and Montha definitella in the clinal zone, we have measures of potential and realized fitness. So the number of fruits before Montha definitella infestation would be a potential fitness, and then realized fitness would be the number of seeds remaining after Montha definitella herbivory. So we have those sort of measures both before and after fertilization, and then before and after herbivory. And we can use that to calculate selection coefficients for populations, both in the clinal zone and in those field experiments on linalool and on other floral traits as well. So the results for the geographic variation were that we saw no difference in pollination, either visitation by hawk moths or bees in any of between any of the zones. We did see some significant differences in herbivory, such that the clinal zone, where linalool is really variable, has the highest herbivory both by H. hyles lineata caterpillars and by Montha definitella. When we added linalool in where it is rare, it increased the number of Montha definitella significantly. However, it was not enough of an increase in the predation to significantly affect seed set. So we saw that even though there was a clear effect of increasing Montha, there was no difference between predicted or realized seed set in the treated or untreated plants. We also saw no difference in Montha definitella between plants from the linalool or non-linalool zone in our experimental array. And we had meant to score pollination there as well, but we actually had very little visitation, which is quite odd in the system. So we ended up only looking at definitella in the array. And then in the clinal zone, where we are measuring flower set, fruit set, and Montha definitella between chemotypes, there was a lot of variation. There was no like common pattern between populations, but we saw some populations where linalool producing plants had much higher numbers of flowers or fruits or Montha definitella, but no sort of consistency across that range. And we only detected selection on linalool production in one population, Rouse, which was also the only population that showed that same pattern or the same significant increase in Montha definitella with linalool that we saw in our sort of augmentation experiment. And the selection here that we see, so this is the selection coefficient sort of on flower number. And this is, so it's flowers, fruits, and then fruits after herbivory by Montha. And so what you're seeing here is a significant selection coefficient between flower and fruit, indicating either fertilization or sort of resource limitation. So this could be positive directional selection by Hyles lineata because they're the primary pollinator and would be affecting the fertilization, or there's some sort of resource impact that we're really not able to parse out. But we also see that in the same population where the number of Montha also increases when you have linalool, that this sort of positive directional selection is completely negated by the effect of higher Montha. So this is no longer a significant term after the effect of herbivory. All right, so to conclude on selection, we do see variation in herbivory across this range. And we see that linalool increases Montha definitella predation in a non-linalool zone population when we add it, and in one clinal zone population, but only one of our clinal zone populations. So there's no sort of strong evidence or of a strong adaptive value or selection on linalool production from this work. Something that I think we failed to capture, even though we did a lot of field work for this particular manuscript, is the potential for interannual variation and the potential effect of drought in this system. So this is a pretty arid habitat, and in years where there's more rainfall, the reproductive output of Oenothera harringtonii is like much, much higher. Um, and so it's really tied to this sort of spring season rains, and then you have the flowering effect of Oenothera harringtonii, and then your reproductive output is pretty driven by how wet that year was. And so I'm wondering if the impact of increased MAMPA potentially where linalool is rare is sort of augmented to the extent that it might have an actual effect on fitness in years where the overall reproductive output of these plants is lower. So I think it'd be really interesting, though also a lot of field work again, to repeat this sort of experiment in a drier year. Because our year, the year we were out there doing this particular experiment was like almost record-setting rains. The plants had a really wonderful year, and we just saw no effect on fitness. All right. So thirdly, here I'm going to look at inheritance. To do this, I have a breeding experiment component in the greenhouse and then a genomics component. So in the greenhouse, I reared and crossed plants and measured heritability, and then identified mode of inheritance of linalool production, as well as three other terpenoid compounds. And then with the genomic data, I tested for associations between genomic variants and linalool production, or again, these other terpene compounds. All right, so in the greenhouse, I collected scent from a greenhouse population that I had actually started for the paternity analysis part. So these are individuals that were grown from seeds from Rouse. I grew them up to flowering, I took scent, I chemotyped everything on a GCFID. I hand crossed that generation as they were blooming and then let those fruits mature, tied everything off. So I had all the seeds and then grew another generation to flowering, collected scent on that as well. And then I have all of those data to do parent offspring regression to look at narrow sense heritability of variation in emission rates, as well as the proportion of Lin plus offspring from crosses between linalool producing and non-linalool producing parents to look at mode of inheritance. For the genomics, I've been assembling and annotating a draft genome for Oenothera harringtonii. And then I extracted DNA and used whole genome resequencing from chemotyped plants across the range. I aligned all of the resequencing data to my genome draft, called variants with GATK, and then performed a genome-wide association study with linalool production, as well as linalool emission rate, and the same traits for a couple other volatiles with Plink2. The breeding experiment showed that parental linalool production predicts offspring linalool production following a dominant pattern of inheritance. So you seem to need a linalool producing parent to produce linalool yourself. However, the parental linalool emission, does not predict offspring linalool emission rates when the non-linalool producing plants are removed. So what we have here on the top is everyone included, all of the parents, all of the offspring. it looks like it's a significant associate or regression between the parents and the offspring. However, if you need a linalool producing parent to produce linalool, then I think this line is probably being drawn by all of these like 0 to 0 points, right? So you have all of these like 0 to 0 points in the data. And when you take those out, the linalool concentration of the parents no longer predicts the linalool concentration of the offspring. So I feel pretty comfortable saying that linalool follows a sort of dominant inheritance pattern where you do need linalool production in one of your parents to produce linalool, but the emission rate variation doesn't seem to be particularly heritable. You might notice that the proportion of linalool producing offspring is actually slightly lower than would be expected for crosses of heterozygous Lin plus plants. So what I have here is the parental chemotypes. So 0 to 0 is neither parent produces linalool. 0 to 1 is the paternal parent produces linalool. 1 to 0 is the maternal parent, and then both parents produce linalool. And so if linalool is a single loci dominant trait, you would expect you would expect the 1 to 1 linalool producing cross to have 75% linalool producing offspring, or if some of those are homozygous, even higher. And then if you had one linalool producing parent, again, and that was a heterozygote, you would expect 50% linalool producing parent or offspring, or again, higher, not lower, if that's a homozygote. So, what might actually be happening here? These the proportions I actually have in my data, so 25% linalool producing offspring with one linalool producing parent and 50 with two, is a really like unexpectedly clean fit of a two loci dominant epistatic relationship. So in this model, I'm showing two heterozygous parents. So they both have one dominant allele at both loci, and then one recessive allele at both loci. And so this gives a really similar pattern. It's sort of the best fit inheritance model for my data, where you would expect like just over 50% linalool producers from two heterozygous linalool producing parents. And you would expect about 25% linalool producers from, this should be a little a, but from a heterozygote parent and a homozygous recessive non-linalool producing parent. So based on these data, the working hypothesis is that this is sort of a two gene or two locus metastatic interaction. And I also looked at this for a couple of other compounds. So alpha farnesene is a green appley scent that is not super common, but shows up in a good proportion of Enothere Harringtonii plants. And what I thought was interesting compared to linalool is that the other compounds that I looked at have different patterns of inheritance. So alpha-farnesene emission rate heritability is relatively heritable. It's about like a 11% H2 value, but it doesn't show any sort of dominant looking or recessive looking pattern. And then I looked at two others as well, beta ocimene, which is the most common compound in Enothere Harringtonii, and then beta caryophyllene, which is another one of these that can be present or absent. And their variation, both in terms of production, present absence, and emission rate, showed no like significantly heritable component as well. I also looked at the genomic data for the species, and I used a genome-wide association study to test for significantly associated SNPs or single nucleotide polymorphism. So single places in the genome that are different across individuals to see if they associated with either linalool production or alpha-farnesene production or emission rate, since that was also a heritable um, volatile trait. I saw no significant association with linalool production. So if this is a two-gene epistatic interaction, I'm not able to pull them out with this data set in any case. But I did find a lot of significantly associated SNPs with alpha-farnesene emission rate. And the ones that were in annotated gene regions were in DNA RNA polymerase and a thioredoxin domain. So taken all together, heritability of floral scent varies by compound, even within compounds from the same sort of class or family or metabolic pathway. Linalool appears to be a dominantly inherited trait in Rouse, potentially controlled by the epistatic interaction of two genes. And alpha-farnesene emission rates are pretty heritable for a volatile trait, and potentially under regulatory control by an RNA polymerase and thioredoxin. So there would have to be a lot of sort of functional work to confirm that. Some sort of outstanding questions here are, would it be possible to pull out those two hypothesized genes if I either one had sort of improved genome scaffolding? This genome draft is still kind of a work in progress, or higher resequencing depth, I think actually would make a bigger difference. And then the genome-wide association study includes different populations than went into my breeding experiment. So it's also possible that there's actually like population level variation in which genes are involved in linalool production across the range. I don't think it's super likely because we did see that these populations are really well connected, but it's certainly possible. So it'd be interesting to resequence the individuals included in the breeding experiment to understand and try to actually pull out that sort of allelic diversity and variation that we hypothesize is resulting in the breeding pattern I see. And then something that is tangentially related and another work in progress for me is a comparison with terpene synthase genes in Enanthera haringtonii and other Onychraceae species whose genomes I'm also assembling as sort of a side project from this PhD. So altogether, what can describe this geographic pattern of variation in linalool production we see in Enanthera haringtonii? Well, it doesn't look like there's any evidence for reproductive isolation of these populations. We don't see any sort of difference in pollinators across the range that look like they would strongly drive variation in linalool production. We don't see that these pollinators would learn to forage in a way that would sort of cause this assortative mating pattern, nor are these chemotypes differentially rewarding. What we do see is differences in the levels of herbivory between populations, where the clinal zone, where linalool is really variable, is experiencing the highest load of herbivory. And we see that in at least two populations, the linalool producing chemotype suffers a much higher infestation of one of these herbivores than the non-linalool producing chemotype. So is Montha definitella predation maybe driving the loss of linalool in the non-linalool zone? I certainly don't have this strong evidence to claim this, but I think it's something that's still worth diving into a little bit more, especially with the idea of how drought might affect reproductive output in years where these plants don't do as well. Does Montha have enough of a negative impact to change fitness between these two chemotypes? It's possible. But regardless of the impact of Montha definitella herbivory, this is an inheritance system that favors the persistence of polymorphisms. So again, from the breeding experiment, the best fit inheritance model would be a two-gene dominant epistatic interaction. And so even if you're selecting for or against linalool production, you're maintaining pretty high allelic diversity of both of those chemotypes in either case. So if even if there's strong selection for or against linalool, you're going to retain a lot of different allele combinations, and you're going to have variation in your offspring generations. Um, it's going to be very hard to sort of completely eliminate or completely sweep linalool across this range. Okay, with that, I'd like to thank all of my funding sources, especially the Society for the Study of Evolution. The Rosemary Grant Award was really helpful in getting all that resequencing data collected and processed and sequenced and analyzed. And I will take questions.
Wonderful. Thank you so much, Haley, for such an engaging and extremely clear talk. Really cool to hear the story of the interaction between these Enothera, the Hyles, the Montfa, the climate roles, etc., and a really beautifully complete data set. Just an absolute joy to listen to. Love to take questions from the audience. Are there folks who have questions? I can get us started with one and get the ball rolling. So, why might the hawk moths not have learned that linalool is linked to a reward? If I recall correctly, Hyles can detect linalool in the antenna, but maybe I'm forgetting that.
No,
Do you have any
they certainly
ideas?
can. Yeah, so we did electroantennogram recordings of both Hyles and Montha, with racemic mixtures of linalool, with each enantiomer of linalool, and with petals of linalool producing Harringtonii. And they know when linalool is a signal. So why they maybe don't, or why the Hyles maybe don't care about it in the field, I'm not sure. These plants are super attractive to them. It's really clear, both sitting out there waiting for them to come as night falls, as well as working in the greenhouses or wind tunnels. You put a Harringtonii plant in front of Hyles and they're like, I want this. It doesn't matter if I smell like linalool or not. And so linalool is not the only volatile in the system. They make a lot of trans-beta ocimene, as well as cis-beta ocimene. They make some like jasmine lactones. They make a lot of other mono and sesquiterpene products. So I think as long as there's some sort of scent signal, it doesn't seem to matter if linalool is part of it. But it is an interesting question because it's really common in hawkmoth pollinated plants.
Awesome. No, that makes sense. I sometimes have wondered about a lot of the work that we've done as well in floral volatiles is one can identify, as you say, a volatile that's important for a pollinator, but necessity versus sufficiency is always a really
Right.
big question. Excellent. Are there other questions? Sounds like Howard's got a question or Amber.
I can go.
Go ahead, Amber.
Okay. Um, Apologies if you covered this, Haley, but how is the linalool produced? I mean, could there be some kind of difference in the environment in these different regions that explains the production of linalool that is just kind of based on where the plants are growing?
Yeah, so linalool is a monoterpene alcohol. It's synthesized by a linalool synthase enzyme. So as part of like the final step in the isoprenoid or terpenoid biosynthesis pathway. And so we have found that even non-linalool producing plants have what look like functional genes expressed for linalool synthase and or genes that would then produce the enzyme. Um, there, the previous transcriptome work focused on a population DC, which is also it's a non-linalool producing population. It's also where I did the augmentation experiment, and then flow, which is all of those plants make linalool every year, all the time. And it was unclear based on the transcriptome data if the variation they were seeing again was sort of like just sort of population level variation, or if it was could be causal, which is why I went back in with the nuclear data or the genomic data instead of the transcripts. But they all, I think, have the hardware to produce linalool, so I expect it's sort of um, uh, regulated at the expression or maybe even protein level, because you could turn linalool into other things as well. It oxidizes quite well. There's a lot of oxidation reactions that could be the next step. And we do see linalool oxide occasionally, but I'm not sure that we've seen just the oxide and no linalool. And then in terms of like uh, abiotic factors, it is a little drier and hotter where there's no linalool. We also looked at soil chemistry. There wasn't any sort of clear distinguishing pattern. So I'm not sure if drier and hotter is enough to change your volatile profile in this way, or if it's sort of like accelerating the impacts of anything else affecting linalool production because it makes it harder to make more seeds every year, basically.
Cool. Howard, I think you had a question as well.
Sure, yeah. I enjoyed the talk. Um, I'm curious, as someone who's worked on chemical communication in insects, very different, and I know nothing about plant pollinator, but is it possible that, especially in relation to selection from pollinators, that what they get is an overall scent? Hence, selection on any one there may be a lot of correlational selection out there that the amount of linalool you produce in terms of attractiveness may depend on how attractive that makes you or not, may depend on the suite of other chemicals you're producing. That is the overall blend that's generated. And if you pull out a univariate trait out of a suite of traits for which there's a lot of correlational selection, you can get results that seem bizarre. And so I'm wondering, is it possible that this is a multi that there's a multi-trait system here with a lot of correlational selection, and so you miss that, you fail to capture that when you try to extract a single trait.
Yeah, I think that's certainly maybe even probably the case. So I I can't find any strong correlations between any of the compounds in or across populations. Um, there, the sort of total volatile emission profile, like rates, are super variable, and then they are usually either ocimene or linalool dominated, but there's not completely a trade-off. It's like if you have a linalool dominated scent, you still have enough ocimene that it doesn't sort of tease apart as a strong correlation between the two. And then the other compounds that are in the flowers, there's like 35-ish, aren't particularly strong signals with the exception of occasionally farnesene and caryophyllene. And so I think maybe what's happening is, again, there's not sort of a like perfect compound or even maybe number of compounds that the hog moths need. It's sort of like there are a lot of compounds that will do and that hog moths find attractive, in combination or not potentially. But there's also, I think, maybe some sort of covariation with other morphological traits. Again, this doesn't come out like across the range, but it comes out in individual populations where, like in Rouse, for example, we see linalool producers are a little bit larger. The corollas are a little bit larger. I think in Berwyn, they have much longer filaments for some reason, so the pollen is like higher up. And then in a couple other populations, there's some variation between like floral tube flare and not linalool, but a couple of other scent traits. And so I think it might be that the pollinators are having a higher effect on the morphology of the flowers, and that might be sort of inadvertently shaping some of the volatile compounds, but I haven't really tested that in a
Like some
robust way.
multivariate selection analyses. Well, yeah, one interesting thing we found in several species of Drosophila is that the absolute concentration of compounds isn't always a very good predictor as to their importance to as a chemical signal. That some very low abundance compounds can sometimes have disproportionately strong roles. Um, it's
Yeah.
just something to keep in mind. But well, it's interesting. Yeah, that if you want to start exploring correlational selection, your data set requirements go up enormously because the models are huge number of parameters you're trying to estimate there, and
Right.
becomes very data intensive in terms of field work. That can be challenging, but
And it can
maybe
be
something
hard.
to consider.
Yeah, some of these populations they're really small. They have like 30 individuals one year to the next. So it's really hard to sort of have the
Get a
power
lot of
to use
replicants. Yeah.
both, right? Like just if I want to look at patterns in the whole region, I end up having to lump if I want to include all of the different traits. So yeah.
Interesting. Thanks.
Yeah, something I think not often appreciated is the sort of overlap between the amount of power you need to do these selection experiments, especially Landy Arnold multivariate selection experiments, and the amount of time and effort and money you need to do field work. So I know there's been some recent work on multivariate analysis of selection and floral scent in particular, which I think Haley, you might have been involved in, possibly. Um, Correct me if I'm wrong.
I've certainly been discussing it with a lot of folks.
Yeah, but I agree, it's really challenging. I've had the same problems myself. I did actually have one question about linalool. I wondered if the presence of linalool might reflect historical selection for linalool that we're not seeing the agents of in the present day. So, or that the agents selection are no longer selected on linalool but had in the past. What do you think about that?
Yeah, something I didn't talk about at all is the fact that this species is believed to be uh, sort of a budding speciation event from Oenothera cespitosa marginata, which occurs higher up in the mountains, sort of like above and to the north, mostly of where Harringtonii lives. But marginata also has like a much larger range. It's a much more common species, and in where linalool is really common is where they're a lot closer to the Cespitosa populations. And Cespitosa marginata is like always a linalool producer. So I think it could reflect the sort of ancestral state of Harringtonii, or there might still be a little bit of gene flow happening in that part of the range. Cespitosa is generally visited by Menduga quinculamata more so than Hyles, but there's some crossover. Um, they're similarly sized hawk moths. And so I think that's something I didn't get any sort of chance to test. But I think there's some potential that linalool reflects the sort of ancestral trait and is maybe being reintroduced to the gene pool from a little bit of influx from Cespitosa marginata.
That would be so cool. Is there any genomic or genetic data available for Cespitosa
There
marginata?
are
Or
like a million leaf samples in the freezers at CBG,
yes,
but so far no one has extracted or sequenced anything.
that sounds really cool, though. Looking at integration between those two in your copious spare time and funding, I'm sure would be really neat. But perhaps a project for the next PhD student.
Yeah.
Uh, are there any other questions from anyone else in the audience? Don't be shy. I think the discussion we've been having so far is really exciting. So,
Yeah, I had something similar to Howard's comment because we see in the lab when we test these olfactory receptors that there can be no response from a ligand, and then the response will greatly increase when another ligand is added. And that other ligand can be at a very low concentration, as Howard was saying, not even be prominent to increase the efficacy of that or response that that other ligand creates. Um, so when you put the moth in the tunnel for the choice or had that choice experiment, that was just linalool in the versus a control.
So it's a linalool being added, but the flowers themselves are they're real flowers, and so they're producing other compounds. And so the reason we did that instead of just growing like linalool producing and non-linalool producing plants. The reason I was experimentally adding the linalool is because I wanted the background scents to be as similar as possible. So in the greenhouse, I grew a whole bunch of non-linalool producing Harringtonii for that particular experiment. And then we didn't chemotype all of them, but we did a subsample, and they have pretty similar profiles. So then we added linalool to that background instead of like just adding linalool to like a paper flower or something like that, or having potentially different linalool producing and non-linalool producing from different populations.
Okay, so the whole plant was in like that wind tunnel chamber. Okay, I misheard that. And then, so sorry about that. And then my other question, or one of my other questions was, instead of having with the variation in linalool production, can you, instead of lin minus lin plus, can there be like high lin, low lin, middle lin, and then like apply that to say the SNP data or the other things? Yeah.
Yeah, yes, actually, it
Mhm.
it there is pretty clearly like a few orders of magnitude range of linol production, and there tends to be almost discrete like lin high, lin low categories. And
Mhm.
so what I did with the GWAS was I didn't bin it like that, and maybe I should have, but I have either plus or minus, or I have like normalized actual emission or concentration rates. So I do have it in there as a quantitative trait instead of like a binomial,
Mhm.
but I never did the three bins. So I didn't find any any SNPs with a quantitative
Mhm.
regression. So I think that's kind of why I didn't try anything else. But
Got it.
yeah, it's a good idea. Might as well look.
Got you. Um, I guess I had one other small thing. Is the idea that the humans are dispersing down that highway, and that's why there's a mixture?
Maybe, but it's usually it's water. So
Oh, okay.
these seeds, they're just gravity dispersed, and the fruit is not fully dehiscent, so it like lets the first half of its seeds out, then it holds on to all the rest and just sort of falls. Um, and so they don't go very far until you have a rainfall event, and then you'll see it come down either like natural waterways, or we see it really commonly on sort of highway roadsides because it likes the disturbed environment, and it can follow that sort of downstream of the highway. And so those clinal populations actually, I think, are a really interesting place to ask questions because they're really, really young. The highway is like only 100 years old, if that. It's probably a little bit less than that, but there have been roads there. So I think that those populations are really young, and there might be a a lot of change happening pretty quickly in that area with these really little populations. Yeah.
Makes sense. Thank you. Thanks a lot for the great talk too.
So very cool. I think we've reached the top of the hour,
Yeah.
and so it's time for us to wrap up. But let's all give Haley another round of applause for an excellent talk and a lovely discussion afterwards as well. And also congratulate Haley on just finishing their PhD. Yeah.
Thank you.
I
And
will
thanks so
just
much, everyone, for coming.
I will just say one or two things just because of the last Greg seminar. Just thanks everybody for the support for the past 3 semesters. I think we've had like 35 or 36 seminars now. And a huge thank you to Katie, who's here, Katie Moore, for making all the flyers and the website announcements and the email subscription that goes out and all the social media and all that. So huge thank you. And we don't know if this will be the last Greg seminar. There may not be any moving forward, but we just want to thank everybody who's been here over the weeks and all the presenters. So I just want to say that. So
Thanks very
thank you
much,
guys so
everyone.
much. And
Mhm.
yeah, we'll see you. We'll see you whenever next time is. So,
And thanks to you too, Robert,
oh, yeah.
for overseeing all this and organizing.
Thanks, Amber. Thanks so much for attending all the and it's been a real joy from my perspective learning about all these different systems. And you never know what's gonna come up next week, plants, animals, whatever. So thanks everybody.
Thank
Thanks
you.
so
See
much.
ya.
Mhm.